Okay, it's 1 o'clock. Let's get started. Thank you everyone for coming to this month's webinar series. I just want to do some quick introductions. My name is Derek. I'm a business development manager here at Genobiotexas. Thank you for everyone for coming today. The title of our talk is Personal Human Immune System Mice, etc., Accelerating the Translation of Immunotherapies from Bench to Bedside. Before we get started, I want to quickly introduce Genobiotexas for those who may not be familiar. So we're committed to advancing biomedical research by providing innovative and high quality animal models and CRO services. So our goal is to help bridge the gap between basic research and clinical application. So after today's webinar, we'll have a brief Q&A session at the end. And if you have any questions after that, feel free to email us and we'll make sure to get back to you as well. So I'm thrilled to introduce our speaker today, Dr. Jing Huang. Dr. Huang earned his PhD in 2004 from the Institute of Zoology at the Chinese Academy of Sciences. He then did his postdoc at Peking University Health Science Center. He has dedicated more than 15 years to groundbreaking research in the United States at prestigious institutions, including the Albert Einstein College of Medicine, the Aaron Diamond AIDS Research Center, Rockefeller University, and Columbia University. As a member of Sigma Xi, the Genetic Society of America, and the American Geriatric Society, Dr. Huang has authored more than 30 peer-reviewed papers in esteemed journals such as Science Translational Medicine and Frontiers in Immunology. His research over his career has focused on tumor immunology, vaccines, infectious diseases, and animal model development. In 2019, he founded Genomab Biotech in New York, where he actively serves as the CEO of the company. And in 2021, he expanded his vision internationally by establishing Nex Vivo Biotech in China. Without further ado, Dr. Huang, the floor is yours.
Thanks for the introduction from Derek. So I'm very happy to meet everybody from here. So now let's begin our seminar. So then I'll close the video. I'm very happy to see everybody from here. So thanks to Genobiotix for the kind invitation to speak today. I'm truly honored to be here. In this presentation, I'll discuss how personalized human immune system mice are revolutionizing the development of immunotherapy, bridging the critical gap between preclinical research and clinical applications. These advanced models are paving the way for more accurate, efficient, and personalized therapies, and I'm excited to share our progress and insight with you. The Genomab is a collaborator of Genobiotix based at Harlem Biospace in New York City, a hub for biotech innovations. So we specialized personalized humanized mice and fully human immune system mice, which critical for immunotherapy research. We also can develop customized humanized mouse models and offer licensing program to support academia institutions, CRO, pharmaceutical, and biotech companies. So our goal is to accelerate the translation of groundbreaking therapies from bench to bedside. So now let's discuss what's the role of these models in studying immunotherapy research. It's well known the human immune system mice have broad applications in translational medicine and immunotherapy. They are used to study antibody-based therapies, cell-based therapies, human-specific infectious disease, autoimmune disease, and human-specific vaccine development. This model provides a unique platform to evaluate the therapies in a human-like immune environment. Then we will examine how humanized mice promote a traditional PDX model for studying anti-tumor immunotherapy. Humanized mice are transforming traditional PDX models by adding a functional human immune system, enabling more accurate study of anti-tumor immunotherapy. This model will allow us to test therapies like bispecific antibodies, immune checkpoint inhibitors, vaccine, cell-based therapies, oncolytic virals, and combination treatment in a human-like immune environment. By mimicking human immune response, this model provides critical insights into therapy efficacy and mechanism, accelerating the translation of immunotherapies from the bench to bedside. Then we'll discuss the challenge in these models and follow and how we are working to overcome them. Developing humanized mouse model that fully replicate the human immune system is more complicated task. One major challenge is to achieve a complete and balanced reconstitution of key immune cells like T cell, B cell, NK, and myeloid cells, which is critical for accurate preclinical studies. Another burden is actual matching between patient-derived tumors and CD34 cell sources, which is essential for personalized immunotherapy research, but extremely difficult to achieve. Additionally, where cord blood CD34 cells are effective, adult-derived CD34 cells are often engraft poorly and heavily regulated to access. Scaling up single donor-derived humanized mice for large-scale study also remains a significant challenge. So finally, the choice between orthotopic and subcutaneous PDX model can lead to distinct results. Will complicate our data interaction and highlight the need for standard protocols. So next, we'll explore how we are addressing these challenges to create more reliable and scalable humanized mouse model. Creating a mouse model with a fully human immune system will involve multiple critical steps. We begin to developing needs to support the engraftment of differential human hematopoietic stem cells to enable robust immune response. So we also need to restore the structure of mouse lymph nodes and B cell germinal centers. Another essential step is to ensure the express of human MHC molecules in the thymus, which is critical for the proper T cell education and function. We need to further enhance the model by introducing a human cytokines in the in vivo, which supports the reconstitution and differentiation of immune system, improving the functionality of human immune cells. So finally, uh, we optimize the engraftment efficiency by using CD34 cells from various sources, such as mobilized blood, bone marrow, cord blood, fetal livers, even iPSC-derived CD34 cells. Together, this step enabled creating a highly functional humanized mouse model so, through their uh, uh, though they are require intensive genetic modification to immunodeficient mice, which can be technical very challenging. Mm, Subsequently, we'll explore how advanced genomic editing protocol is for further enhance this mouse model. It's well known the NOG and NH mice are widely used for engrafting the human immune system, but genetic manipulation on their NOD background is very challenging. Traditionally, this requires backcrossing NH mice with the transgenic C57 mice, a process that is both time-consuming and lab-intensive. At the genome, so we collaborate with the GenoBioTX, developed advanced genetic approach to simplify this process using CRISPR-based gene ablation, knock-in humanizations, and microinjection-based gene expression. We can directly edit NSG-similar mice without extensive backcrossing. So this significantly reduces the time and effort required for genetic modification on the NOD background. These innovations allowed us to create more sophisticated and humanized mouse models faster and more efficiently. Then we'll dive deeper into several mouse models and their unique features. The NVG mouse model is similar to NSG and NOG mice, providing a robust platform for human immune system engraftment. Building on these models, we have developed NOGF models by using CRISPR technology to knock out a special gene in the NVG, which significantly reduced the number of bone marrow cells in the host. This modification will create an even more permissive environment for the engraftment of human immune system or the patient-derived xenograft, outperforming the traditional NSG and NOG models. To validate this, we conduct experiments involving the engraftment of human PBMC. So in our experiments, NVGF mice show nearly about twofold higher PBMC engraftment compared to the NSG and NOG mice when inject the same amount of PBMC. We then tested the anti-tumor effect of PD-1 antibodies in both PBMC engraft NVG and NVGF mice. Surprisingly, the PD-1 antitumor effect only observed in NVGF mice. This suggests that the reduced bone marrow in NVGF mice not only enhance PBMC engraftment, but also minimize the interference from host immune functions, such as ADCP, that can inhibit human T cells. While these results are promising, we need to be validate with more donors to ensure the consistency. The next, we'll explore how we can further improve these models by recovering lymph node function. The lymph nodes are essential for the immune response, but the development is compromised in NSD or NOG mice due to the autoreceptor gamma chain dysfunction. To address this, we developed NVGT and NVGR mice by overexpression of TS3 gene using advanced microinjection technology and humanized LR gamma T using CRISPR technology in the NVG mice. These modifications restored lymph node development, creating a more functional immune system that supports the antigen-specific adaptive response. While these results are promising, further study need to do to fully characterize the impact of lymph node recovery on immune response. The next, we'll discuss T cell development in human immune system mice and their implications for immunotherapy research. In the classical immunology, the thymus is critical for T cell education. However, in NSG mice, the thymus is compromised in the neurals and shrinks, becoming non-functional in adults. So it also lacks HLA expression, limiting the T cell development. To address this, we develop NVGA mice by replacing murine B2M with human HLA B2M fusion gene. This allows HLA-A2 expression while disrupting murine MHC1 function. Unlike Jackson Labs' NSG-A2 mice, NVGA mice enable HLA restricted T cell response without murine interference. Our hypothesis is that after initial T cell priming in the thymus, human antigen-present cells may further promote T cell development. The next will explore the additional modifications needed for a fully functional human immune system. The development of fully human immune system in the mice requires human cytokine to support the differentiation and function of various human immune cell subsets. The NVGF-TA mice represent an advanced model that combines reduced host immunity, restored lymph nodes development, and actually restricted T cell education with the expression of different human cytokines. This combination creates a more robust functional human immune system, making NVGFTA mice a super platform for studying human immunology and immunotherapies. Subsequently, we'll explore the role of human cytokines in human immune system in human immunity development and how they enhance this mouse model. Human cytokines are required for the developing different subsets of human immune cells in humanized mice, as we show in the left figure. However, their inclusion requires careful consideration. So we must identify essential cytokines for human immune system reconstitution and ensure that they do not cross-react with the murine immune system. It's also critical to optimize cytokine expression levels, durations, timing, and tissue-specific control to actually introduce a functional human immune system. We must also consider the competition and synergy between the cytokines to avoid unintended efforts. Finally, the cytokine combination should be tolerated to specific research topics, such as cancer immunotherapies and infectious disease, autoimmune disease. Based on these considerations, we express more than 5 cytokines long-term to introduce a robust human immune system. So next, we examine the phenotype of human immune system development with the aid of this optimized human cytokine. HSNVG plasmids engineered with optimized human cytokine expression show enhanced human immune system reconstitution compared to the standard NVG mice. They displayed higher engraftment of human CD45 cells, CD33 amyloid cells, and NK cells, indicating a robust and functional human immune system. These mice also have extended lifespan to up to 30 weeks post the infusions, outperforming the models like SGM3 mice, about 16 weeks, and NOGEXR mice about 25 weeks. These make them ideal for long-term study. The next will analyze the spinocyte subsets to further validate their function. Through intensive flow cytometry analysis, we have identified a complete human immune system in the spinocyte of CD34 NVG plasmids. These include rare subsets like Tregs, MDSC granulocyte dendritic cells, and M2 macrophage, which are often absent or underrepresented in conventional CD34 humanized mouse models. This finding highlights the robustness of the NVG plus model in supporting diverse human immune cell populations. Next, we'll explore how expressing these cytokines in NVGF mice, which have reduced bone marrow cells compared to NVG, further enhance the human immune system development. By expressing more than five cytokines in an open niche environment, NVGF plasmids demonstrate a significantly higher frequency of human myeloid cells and NK cells in the blood compared to the Jackson Labs newly released NSD-FLT3 interleukin-15 mice and NSD-STM3 interleukin-15 models. This highlights the superior ability of VDF plasmids to support these critical immune cells populations, which are essential for studying immunotherapy and immune response. The next, we'll take a closer look at these cells at the transitional with the additional markers at 30 weeks post CD34 infusion to further characterize their functionality and longevity. At 30 weeks post HSA infusion, flow analysis of NVDF plus mice revealed a surprising population of Tregs and PD-1 positive CD8 T cells, which are not high enough in other models. This makes NVDF mice ideal platform for studying immune checkpoint inhibitors and immunosuppressive role of Tregs. Further analysis will explore additional markers on lymphocytes, such as memory T cells and B cells, to fully characterize this immune profile. Uh, Next, we'll examine the subsets of myeloid cells to understand their role in the advanced humanized models. The flow analysis NVGF plasmids at 30 weeks post HSC infusion detect myeloid-derived suppressor cells, granulocytes, indicating this model with potential for studying the tumor microenvironment. While dendritic cells are rare in the blood, they have a significantly detectable in the bone marrow and spleen due to the timing issues. We'll show bone marrow findings in the future discussion or after the seminar. The next will examine the engraftment of human immune system using adult CD34 cells. Conventional CD34 humanized mice use cord blood or fetal liver-derived CD34 cells, which create a human immune system but fail to reflect individual immune response or personalized therapy outcome. Recognizing this limitation, leading labs, including Jackson Labs, Yale Universities, and Columbia University, have begun explore adult CD34 cells. However, the engraftment of these adult CD34 cells, especially mobilized CD34 cells, is significantly lower compared to the cord blood. The next will showcase our breakthrough in generating CD34 cells using mobilized adult CD34 cells, which overcome this challenge. To improve the engraftment of human immune system using mobilized adult CD34 cells, we infused MB34 cells into NVG plasmids and NVGF plasmids. These mice show that NVGF plasmids have a significantly higher CD45 and T cell development compared to the NVG plasmids, indicating more available niche, and the ability of optimize the cytokine combinations to improve adult CD34 cell engraftment. This is a major advancement, as adult CD34 cells are notoriously difficult to engraft in NSG or NOG mice, even with the cytokine support. Since mobilized adult CD34 cells can be isolated at a large scale from healthy donors with detailed array information, a single donor can generate uh, hundreds of humanized mice with the potential array matching to the PDX models, enabling the antigen-specific immune response. The next, we'll explore the development of his mice using iPS-derived HSC cells. The adult CD34 cells isolated from the bone marrow or cytokine mobilized PBMC face the ethnic or practical challenges, including limited availability and heavy regulation to access these clinical samples. IBS cell-derived CD34 cells offer promising solution, but successful engraftment has not been documented. However, in our preliminary data show detectable human immune system engraftment in NVGF plasmids, making using the iPS cell derived CD34 cells. So while these results are very exciting, they need further validation through repeat experiments and more robust data. The next will explore how this breakthrough can revolutionize personalized immunotherapy research. Personalized humanized mice, particularly those use adult CD34 cells, are invaluable for evaluating personalized immunotherapies and studying individual immune response to the drug. In the future, steps will be taken to uh, revolutionize drug development by combining personalized humanized mouse models with AI to predict clinical outcomes and optimize therapies. This highlights the need for more partnership in this field uh, with different labs and companies. The next will summarize our key technology in our mouse models. In this slide, we compare the Genomap's personalized CD34 mice and the conventional CD34 mice, highlighting the advancements in our models. By combining reduced host immunity, optimized cytokine expression, we can develop fully human immune system mice using different sources of CD34 cells, with the key improvements including higher engraftment efficiency, better immune cell reconstitution, and support for diverse immune cell subsets like Tregs, MDSC, NK cells, and dendritic cells. These advancements position Genomap's personalized CD34 cell mice as a next generation tool for translational research. Our mission is to enhance the translational accuracy by developing advanced humanized mouse models that bridge the gap between preclinical research and clinical practice. The key goal include in graph generating single donor HSC mice with actually matching to the PDX samples, produce cost-effect models at a large scale using optimized human cytokine, and creating customized mouse models tailored to diverse research needs. We also aim to develop humanized mice from various CD34 sources and apply them to test the immunotherapies and model immune-related disease like infectious disease and autoimmune conditions. To achieve such goals, we need a strong collaborations across the academia, industry, and biotech to drive innovation and improve the reliability to translating lab findings into clinical applications. Now, I would like to open the floor for questions and discussions. Thank you very much.
Thank you, Dr. Huang, for that great talk. So if anyone has any questions, feel free to put them in the chat and we'll be sure to answer them. And again, if you have any questions you think of after this, feel free to email us as well.